His cartoons from his published papers. Those who know Prajwal and those who don't know him as well always ask the question, where is Prajwal? And you'll notice some of these stamps, Evanston, Cambridge, Ghana, even the beach. Well, Prajwal first came to Northwestern in 2002. Was an undergraduate, initially and through the honors program in medical education. Along the way, really discovered science, enjoyed opportunities to work in laboratories, was involved in many activities, public activities, civic activities, which is where I think I first ran into Prajwal, various committees. Decided on the medical scientist training program, MD PhD, which, having been very involved in it, I always felt were for the truly crazed. Right, Taylor? I mean, the idea that you were unsatisfied with one degree, you wanted it all. You wanted to be a discoverer, and you wanted to see that your discoveries would impact on humankind. And of course, along the way, Prajwal also was really involved in the humankind component, really interested in human healthcare around the world. Chris Dobson, a dear friend and colleague, the master of St. John's College, their seats up here, at the University of Cambridge and Department of Chemistry, and I would talk a lot, wonderful ideas of science. And like a lot of things that happen to scientists when we go to meetings, those ideas never come to completion. There's no fruition. Because they're ideas, there's no one to do it. Which led to, where's Prajwal? Prajwal comes to join the laboratory. I mentioned to him, well, there's this dear friend Chris Dobson in Cambridge. I think this could be a perfect match. Chris being a physical biochemist, our laboratory being a biologist, laboratory with common interests, we needed someone who could translate. Prajwal then goes to Cambridge, as you know, to St. John's. Along the way, gets a Fulbright fellow position in the UK, ends up at St. John's. There is evidence that Prajwal is at St. John's. Some of you who are wondering, was he even there? And of course, St. John's in Cambridge, as Bob Lamb and many others know, is an absolutely gorgeous, the River Cam, hunting along there. The portrait of Chris Dobson, the master. And of course, some of the enjoyments of being at Cambridge and St. John's, the May Ball, of course, held in June, wonderfully. I don't normally wear a tuxedo. Those who know me, I barely will even wear a jacket. But for this event, for Prajwal and for Chris, I in fact even showed up with a tuxedo. And we had a wonderful time, of course. This is what it takes for me to see my own student. Anyways, it's really a pleasure. Having Prajwal in the laboratory has been a singular experience. And he's really, I think, as you'll see in this seminar, really contributed greatly in a way that I think only he can. With that, Prajwal, welcome. And for those of you who are not as familiar, he is now Dr. He did receive his PhD from Cambridge Chemistry earlier this year, and successfully and wonderfully defended his neuroscience PhD at Northwestern last week.
Something like that.
Something like that. And of course, this is his public seminar to friends and colleagues. Prajwal.
It is absolutely wonderful to have all of you here with me, and I'm very grateful to everybody here and for contributions large and small, to my own small contribution to our thinking on some of these problems. And I'm excited to share with you, including some people who are distant in Bangalore and Washington and downtown Chicago as well. I hope you guys can sort of see me. You shouldn't see anything on here yet, so that's okay.
Is this being on all the Megatrons in lieu of the World Cup?
Absolutely. They are not in the World Cup, so they're not particularly interested. There's no cricket match going on right now. So this is all the entertainment they've got, so I'll try to be entertaining. And so I'm very excited to share with all of you, friends and family, a little bit of what I've been working on the last few years. Some of you have been wondering, why is it taking me so long to finish school? Yes. And so I'm very happy to try to convince him that maybe it was sort of worth the time. But our story doesn't begin 6 years ago. It begins about a little over 100 years ago, when Augusta Dieter walked into a hospital in Germany and told her doctor, a guy named Alzheimer, that she thought she had lost herself. And Alzheimer wasn't the first person to see the crippling effects of dementia on individuals and families and society. Of course, people had seen their parents and grandparents degenerate for a long time. But he did start to ask a really fundamental question that had only recently become possible to ask, which is, what is underlying the profound and devastating changes that someone like Augusta Dieter experienced? How had she lost herself? What was the biology of that? And he characterized, over the following few years, these extraordinary and disturbing plaques, these clumps of protein that riddled her brain and the brains of other patients with Alzheimer's disease. This is his drawing, and this is the actual slide with this very dense proteinaceous material. And what he speculated at the time, there was no way to know. They had no idea really what was in there. They barely had an idea what proteins were. It had only been in the last few decades that heated debate over what a protein, this fundamental molecule for life, what even constituted it. You know, the year before Augusta Dieter walked into their clinic, Emil Fischer had made the first convincing argument of what the bond that connected individual monomers in a protein even was. So this was new days. But there was a sense that whatever this was, it had a profound connection to the memory loss and the loss of self that this patient and many others experienced. Now, the question that inspired, and that really people have been trying to answer with respect to this disease and many others, is how the extraordinary complexity of such a human disease, the complexity in the way that it affects people's lives, connects to the remarkable complexity of cellular biology, which we're still trying to piece together and sort out. What is that connection? Where can we find it? And there are two, I think, big ways of thinking about this. Either this complexity multiplies on top of each other and makes this a total mess that we'll never be able to sort out. Or maybe there are principles and rules that we can work out that will, to some extent, make it easier not only to understand these diseases and understand this biology, but find ways to diagnose and treat it and treat these diseases in the future. And I'm an optimist, so I hope it's the latter, and I think it might be. So then, this talk really is about rules. Rules for a collapsing proteome, rules for collapsing cells, rules for people whose lives are collapsing, and how we can use a concept called metastability that we'll go into to try to understand the dynamics and changes that, that cells experience in health and disease. Now, a week or two ago, Taylor talked about metastability in exquisite detail and with excellent experiments. And what I'd like to propose is no exquisite detail and no excellent experiments, but rather lots of very broad efforts to model with computational approaches thousands of proteins at once instead of doing a good experiment on one protein like a sensible person. And of course, as Rick mentioned, I've been bouncing around between these 3 labs for the last, well, 6 years. I know longer than Mom and Dad you were hoping it would be, me too. But I've had a terrible amount of fun, and I'm excited to share that with you. Now, we already talked about Augusta Dieter. These diseases, you know, it's compelling and it's powerful to talk about one person. But we have to remember that, in fact, it's Alzheimer's, just as one example of a neurodegenerative disease, is almost epidemic. 25 million people affected worldwide. That number is expected to quadruple. The biggest risk factor for Alzheimer's is aging, and the population is aging rapidly. And right now, the caregiving costs for Alzheimer's, just in the US, $172 billion. Keep in mind that the entire healthcare spending in this country is about $3 trillion. And one disease is contributing 5% of that. It's massive. And we don't have a good treatment. So we spend all this money and people and their families are still suffering. And so then the question is, where can we look for the origins of the disease? We got to look deeper than the outward affect of the patient, of course. Probably the clue is somewhere in the person's brain, the hippocampus maybe, where memory is encoded. Maybe we can look deeper at the neurons. But then eventually we need to ask, what's going on with these proteins? How do the proteins that are implicating a disease like Alzheimer's, and we'll discuss Parkinson's, Huntington's, ALS, and others as well, how do they go from being their normal shapes to ending up in those big, massive clumps that we saw before? And they get there, and especially because I know that a lot of folks here, their expertise lies elsewhere. Just do a quick, recap to proteins. Why do we care about them? Well, they're really the workhorses of life. Most of the things that happen in the cell, most of the chemical reactions, most of the structure that holds the cell together, in some way, proteins mediate that, transferring information and providing the support, structure, functionality that allows each of us to be alive, to be quite frank. And so here we can see an example of that, where these striations are all proteins basically holding together the shape of these cells that we see here. The blue is an outline of the cells, and so they have a profound role and numerous roles, and there are thousands of them. There are thousands of different kinds of proteins that have different shapes, different functions, and a great effort of biologists to try to make sense of what these proteins are doing. Now, they form these, in order to mediate their functions, they need to form shapes of exquisite complexity. If I asked you to make a loop like this with a piece of rope, you'd probably do it pretty easily, right? I figured that'd be not too hard. Alex, could you handle it? Yeah, probably. What about Alex? If I asked you to make this,
Not so easy.
Not so easy. I probably couldn't even do this. This I could do. Proteins, or the cell is manufacturing proteins like this at an incredible rate, on the order of milliseconds and seconds. And the proteins somehow manage to fold themselves up into these functional shapes well enough that we aren't doomed, well enough that they can do all of those functions, and that they don't generate toxic species that could kill you. Well, they do sometimes, that's the problem. But well enough that people live for 70, 80, 90 years. And it's extraordinary. It's almost unimaginable that you can go from the way a protein starts, which is just a little string, and fold up into these complex shapes. If we try to attach some numbers to it, it really becomes clear. Something called Cyrus Leventhal proposed that there's a bit of a paradox here. Even small proteins can occupy this incredibly massive number of possible shapes, 10 to the power of 100, more than there are atoms in the universe. And yet, from all of the possible shapes that this protein can occupy, it finds the one that works in a second, in a millisecond. Alex and I can't even fold one thing together. I definitely can't. And yet, this happens at a massive manufacturing scale in all organisms all the time. It's truly one of the great miracles of life that we all can even manage to be alive, given the magnitude of this problem. And so how would that happen? How would we mediate such a tremendous change, such a tremendous phenomenon? Well, a big part of it is that the chemical principles that, that sort of define this sequence, this little chain, give directions to allow the unfolded species to become folded. And then there are lots of other helpers in the cell to make sure that it happens efficiently. We'll talk about some of them today. Things called molecular chaperones. They are what they sound like. They help to chaperone this process, make sure the proteins hopefully don't get into too much trouble on their way from this state to this state. But even understanding that those chemical principles are somehow operating, even understanding those principles in some detail, we find it incredibly difficult to then work out how this actually goes from here to here. You can take all the computing power in the world and we can maybe model a very short period of time for the dynamics of a protein. It's an incredibly complex problem, but again, one that gets solved all the time. But when it doesn't get solved, we end up with many, many different devastating disorders. We've already talked about Alzheimer's, Parkinson's, Huntington's, ALS, numerous others. There are dozens of what are called protein misfolding disorders, where the folding fails and instead these aggregates, like the ones with amyloid beta and Alzheimer's, Huntington and Huntington's disease, here TTR, which is another much rarer disease where the protein TTR aggregates. And the same is true in almost all of these, that you find these well-defined inclusion bodies that are a hallmark of something going wrong in the folding process. The formation of insoluble, they can't be dissolved in the normal sort of water of the cell, insoluble protein aggregates. This seems to be a hallmark of the disease, and we've known that for about 100 years. But what we've thought for quite a long time is that this is weird. This doesn't usually occur. Very few proteins can actually form these clumps. Most proteins behave well, like myoglobin, which we saw before. But that might not be the case. And in fact, we can go as far back as the 1930s. Bill Asbury, one of the first great crystallographers, he got some protein from a guy named J.D. Bernal, another leader in protein biology at the time. And J.D. Bernal had crystallized this protein, and it was nice, sort of globular shape, well-behaved. And Asbury left it out for a little too long. And so you see in that issue of Nature, there's the crystal structure from J.D. Bernal, which looks like you might expect a protein to look. And then a short note from Asbury afterwards where he says, you know, my buddy Bernal sent me this protein, and I left it out. And I see it not forming globular structures, but I see it forming fibers. And maybe that means that proteins can occupy either shape, that they're metastable. That means they have two different stable shapes. They can either be little spheres, or they can clump up into these fibers, which we've seen other proteins, structural proteins like collagen, or proteins in the hooves of horses form. Maybe there are these two different possibilities. Now, for decades, people thought, okay, that's curious, but what does it have to do with anything that we care about? Well, the sort of aggregation that's happening in Alzheimer's, Parkinson's, Huntington's, ALS, that aggregation may be that generic phenomenon. So what we have here is a much more detailed schematic of what those aggregates look like. What's called beta sheets cross-interact, they intercalate, and you get a cross-beta structure and form a fiber along this axis. And that seems to be highly characteristic of the aggregates we see. And what's extraordinary is that it's not just so one, we've got that the aggregates in the disease form the structure. And then two, it's not just the aggregates in the disease. So take myoglobin. Myoglobin is the classic example of a very well-behaved protein. It was the first protein that was crystallized, which means that it doesn't, so you can put it in a very high concentration, it doesn't start acting funny. You're not going to get an easier protein probably to work with than myoglobin. But Chris's group showed in 2001 that if you kick it hard enough, if you put it under the right conditions, you can even make myoglobin start to form these fibrils, these aggregated structures. So regardless of sort of how well constructed and well behaved a protein is, under the right conditions, it reverts to this almost ancient and primordial state where it forms these fibrils, which are more characteristic of the general properties of proteins than the specific features of the individual protein we're looking at. So at some point, This phenomenon that we see in diseases like Alzheimer's, Parkinson's, Huntington's, this is a phenomenon that is a general principle for the chemistry of the protein molecule. It's a reversion to a fundamental but also a pathological state. And much of protein evolution has been about controlling and avoiding that kind of structure and getting one that's more useful instead. Not only is this aggregation generic, but actually in the context of stress and disease and aging, it's widespread. So with those clumps that we saw before, you have co-aggregation, where other things come and stick to it and misfold. You have independent aggregation of proteins when you stress a cell, when you heat it up, over the course of aging as an organism's ability to manage folding gets disrupted. You have degradation of proteins upon stress when they fail to be made properly or efficiently. In fact, there's an estimate that about a third of proteins, as soon as they're made, are marked for degradation. That the process is low fidelity, you're trying to make as much as you can. You don't worry about the fact that you make a lot of mistakes. You just keep the manufacturing process up. And here's an example from some really exquisite experiments from Rick's lab on the temperature sensitivity and, again, the metastability of proteins in the organism C. elegans. So what we're looking at here is a picture of this worm, 959 cell worm C. elegans, and what we're looking at specifically is we're focused in on a muscle fiber. Now, this muscle fiber is in part composed of a protein that has a special property of being temperature sensitive. What that means is these striations are nice and well folded and formed at 15 degrees. But then at 25 degrees, you see the fibers are all clumped up and broken up. There's been a misfolding event, and these proteins have begun to aggregate instead of organizing the way they're supposed to. But temperature isn't the only way you can make it happen. That paramyosin protein, if you express another toxic protein, so this Q40 is a protein that will clump up and is a model for a class of aggregation diseases known as polyglutamine diseases. If you let this protein clump up, even at the temperature where paramyosin should be functioning, it begins to clump up as well. So it's this idea that disruptions in one part of the protein homeostasis, or what we call the proteostasis network, disruptions in the balance of the cell, can push many different proteins into stress and misfolded states. And it may be that whatever the initial event is, whether it's temperature, the expression of a toxic protein, other stresses, the decline that naturally happens in aging, that any one of those stresses then manifests across the entire proteome, across the entire collection of proteins that characterize the organism and cause widespread and complicated disruptions. There are hundreds of proteins that are implicated in vivo in these diseases, not just 1, 2, or 3, the way we're used to thinking about diseases. And this problem is awful complicated because this is sort of a schematic of the pathway of protein folding and misfolding where things get aggregated or degraded or folded or form crystals. And actually, it's not just happening for one protein, right? It's happening for lots and lots and lots and lots of proteins all at once, and somehow this problem gets solved or it doesn't. And as we try to understand the disease, we're trying to disentangle this. And so when we see the disease, what do we see? This is a network map of the various proteins that are thought to be associated with Alzheimer's disease. Lots of different cellular compartments, the mitochondrion, the endoplasmic reticulum, along the membrane, lots of different pathways, lots of functions, calcium signaling, oxidative phosphorylation, interactions into the nucleus. There's a lot going on here. And the really depressing view is that there's a lot going on. Each one of these things is specific and important, and you need to study each one in incredible detail in order to understand the disease as a whole. And maybe, again, all of this maybe follows a set of guiding principles, or at least much of it does. And if so, could we reverse engineer and work out what exactly are those rules that mean that this set of proteins are in trouble but others aren't? And that really has been the thrust of what I've been trying to do for the last few years, is to make sense of why these complex networks of disruption are evident in a disease like Alzheimer's or Parkinson's or Huntington's. And you can take a sort of general approach to thinking about such complex problems. You can go bottom up or top down. Bottom up, the question is, can you take simple rules to explain complex biological phenomena? So can you say, well, I know that physics and chemistry works like this and this and this. I know there are these rules, that high concentration things are going to clump up together. And can I use that to see if I can explain the much more complicated phenomena that we see in the cell? It's a lot like this great little thing called Langton's ant. It's a little Turing machine that was developed in 1986. And the idea is very simple. This ant moves across a checkerboard, and as it moves, it changes the color from black to white or vice versa, and the direction is also encoded. There are only 3 or 4 rules that govern Langton's ant. But if you let it run out for long enough, you get exquisitely complicated shapes. You can start from the simple rules that the ant follows and get great complexity. I think maybe to some extent the same is true in biology. But this doesn't help that much, because you can start from the rules, but it doesn't give you a good language to be able to explain what it is you're seeing. So can we summarize the biological processes that, say, Langton's ant explains with quantitative terms that sort of go from the top down, that give you a big picture? A great example of this is from anthropology, actually. Something called Dunbar's number. Robin Dunbar observed that the mean group size for organisms, meerkats or bees or people, or probably not bees actually, mammals, meerkats, chimpanzees, people, is correlated rather well with the size of the organism's neocortex. So then what this means is you can take this very complicated idea of group size, of how organisms interact. And either you can say, well, I'm going to study this organism in incredible detail and work everything out about it, or you can say, well, actually, we've got this great summary statistic, which is easily measurable, the surface area of its neocortex. And that tells me a whole lot about what's going on. It's a great way of summarizing, again, succinctly, this much more complicated phenomenon. So can we think about ways of doing that in biology as well? And so overall, this gives us this kind of scheme. Can we find the global risk points for metastability across the proteome? This is basically showing you the life cycle of a protein, starting from the ribosome where proteins are made unfolded. Now the protein can get folded. It could be degraded, like I said, a lot are. Could be misfolded, where it's aggregated or degraded. Actually, it could also be refolded here, left out for simplicity. Even after the protein's folded, it might still be at risk. Now, looking at what happens when the protein is coming right off the ribosome, that's a set of work we've done to predict what's called co-translational protein folding. That's the idea that that folding process goes ahead and gets started while the manufacturing of the protein is still in process. And that tells us a lot about the first moment where things could go awry or could be managed properly. And then the second set, where we'll actually start, is on an idea called supersaturation, which we think is a way of rationalizing a lot of the protein misfolding and aggregation that occurs in stress, aging, and disease. So protein supersaturation is an extremely simple idea, and it's not even really our idea. We can thank Bill Eaton for it at the NIH. But the idea is just that, as with any solution, if you put too much in it, you're trying to make some Ovaltine, mix the grounds in there, you put too much, some of it's going to float to the top. It can't be dissolved. So concentrations are extremely high. Proteins may no longer be soluble. They might be in a supersaturated state. And the interesting thing is that under the right conditions, the grounds may not float to the top right away. They might sort of be trapped in the solution, and just tapping the side of it will make them come out. That could be happening with proteins as well, where they're being maintained at a concentration that's very high. But how do you know how high is too high? Well, that has to do with the chemical properties of the protein. So here, what is the propensity of the protein to aggregate? Or vice versa, how likely is it for the protein to be able to be dissolved? So if a protein is very prone to aggregate, even a small concentration could put it at risk. But if it's not very aggregation prone, you really have to drive the concentration up really high to put it into this dangerous zone. You'll also notice that I keep switching between the English and the US spelling of aging, depending on who got the last word on the figure, whether it was Rick or Chris. One of the great advantages of the fact that protein aggregation is generic and that it's widespread is that it's actually predictable. Because it has to do with the basic underlying principles of the chemistry of the protein, we can use information we know about what's called the primary structure, the assembly of monomers along the protein of amino acids, to make a prediction of how likely a protein is to clump up. And this has to do with how hydrophobic the protein is, what secondary structures does it form, alpha helices or beta sheets, then the charge of the individual amino acids in the protein, as well as the pattern of hydrophobic and hydrophilic residues. And this all comes from fantastic work from Fabrizio Chiti, Giangaetano Tartaglia, Mo Power, Kateri Dubé, and Chris and Michele's groups. And what they've observed is that if you create a profile based on this information, you can actually predict the parts of the protein here, the peptide amyloid beta from Alzheimer's disease, that drive the aggregation process. So this is the predicted profile, and this is where we know the aggregation-prone regions are from experiment. And it turns out they line up pretty well. So we can use this information then to look at thousands of proteins and ask what's at risk and what isn't. As I said, we benefited tremendously from ideas from Bill Eaton on the idea of supersaturation. It's cut off a little bit, but basically, supersaturation is the state where there's more solid, more whatever, your Ovaltine, that's dissolved in the solvent than is possible under normal conditions. So now this is, again, going back to this idea of metastability. It's a state that's metastable. Because as long as you don't do anything and you don't look at it the wrong way, maybe things can stay okay. The second you tap the side of that glass, everything falls apart. It's stable, but not quite. It has two different states it could be in. And this is a general principle of chemistry. You probably learned it in middle school. But the application to proteins is really interesting. Bill Eaton was studying a mutant of the hemoglobin protein that causes sickle cell anemia. Now, sickle cell anemia could in many ways be considered a protein aggregation disease. You have a mutation, it causes misfolding of the protein, and then you form these fibers that disrupt the shape of the cell and cause some devastating consequences for patients. Now, you can think about that formation of the fiber. You can ask, how long does it take before that happens? What's the delay time in the test tube before you see the fiber formation? That's the delay time for aggregation. That is inversely proportional to this idea of supersaturation. So the more of the protein you have around, the shorter time before you see it clumping up. Makes sense. But it's not just the concentration. You also need to, so you need to know what that total concentration is, but then also how much, based on its chemical properties, could be normally dissolved in the solution. And the more you have above that, the more supersaturated you are. What's sort of crazy is that there's this really strong exponential relationship. So here you've got the delay time, and here you have the supersaturation ratio. So the more supersaturated you are, and this is the inverse of the delay time, the shorter the time that you have before you begin to aggregate. And the power on this, these are all in log scales, the power on this is, somewhere on the order of 30 or 40. So just a small change in supersaturation causes a massive change in the kinetics and the timing of the aggregation process. And so we thought, well, if this is true for one protein, why can't it be true for 20,000? And let's see if we can make some rough estimates of that. So we've extended the idea of supersaturation to the entire proteome. And the way we do this is to try to estimate the supersaturation from that unfolded, expanded protein, but then also once the and let's talk about that first. So we've got the total concentration, which we estimate from the mRNA level. So this is the molecule that encodes for the synthesis of new proteins. And then we predict this soluble concentration as the inverse of the aggregation propensity. So the less aggregation prone you are, the more you can be dissolved in a solution. And something very similar from the folded state. So once a protein is made and folded, a lot of the problems that the unfolded sequence has get buried inside, a lot of the problematic areas. So now the folding dynamics are different, and the misfolding dynamics as well. But also, it's not sort of unfolded protein you want to look at. You want to look at the overall concentration of protein in the cell, most of which is likely to be folded. So we have a different estimate of that from experiments that other people did. That's going to be a theme. And then we also make predictions of the aggregation propensity, taking into account that the parts that are problematic might already be buried inside the protein, and so you can avoid some of the problems. And using these two estimates, we essentially estimate the supersaturation for tens of thousands of proteins. And we asked, are the proteins that are associated with aggregation in disease, in cell culture model systems, in aging, are they more supersaturated or less supersaturated than the rest of the proteome? These are the amyloids. So these are the disease-related proteins that form this clumped up structure, which is known as the amyloid structure, and they are exquisitely supersaturated. These are likely to be the driving aggregation-prone constituents of disease phenomena. So you'd expect them to be really high. If they didn't, I would stop talking now because we would be totally off. But so, okay, so these are problematic. What's stunning, though, is that we're not looking at disease states here. These are the normal, we use the normal concentrations and the normal sequences that these proteins have, even in a healthy sample, these proteins are already in trouble. And little changes, a mutation, an increase in expression, could really push it over the edge. But then also, we look at things that co-aggregate with those plaques, for instance. So in addition to amyloid beta, which is most of what you're going to have in this plaque, there are lots of other proteins that essentially decorate the outside of it, that get stuck as they're going along. You might expect that that happens by chance, that they're just innocent bystanders. But in fact, they have these same features of supersaturation. So things that co-aggregate with plaques, with tangles, which are another aggregate seen in Alzheimer's, Lewy bodies, which are seen in Parkinson's disease, and then artificial beta structures, which are very similar to these, but essentially a much more controlled model that was developed by Ulrich Hartl. In all these cases, the proteins that get stuck to these things are more supersaturated than other proteins. And I should say that essentially what people thought before this is, well, okay, you've got these sort of somewhat random lists of proteins that are associated with this or this or this, and maybe there's specific reasons why protein A co-aggregates with a plaque and protein B is seen in a Lewy body. But what we've tried to do is to say, well, in fact, despite the disparate, messy, complicated experiments we have here, there's a general underlying principle. So some of that complexity is getting disentangled, and we can say, well, actually, there's a logic here. And then finally, in the worm C. elegans, we saw some pictures from it before, hundreds of proteins have been shown to aggregate as the organism gets older, just normal natural aging. Those proteins tend to be supersaturated as well. So a lot of different biological phenomena being explained with a similar general principle. So we wondered, okay, what are these proteins and that are most particularly supersaturated, and do they have anything to do with the kinds of systemic collapse that we see in disease? And in fact, if we ask what biochemical and disease pathways are associated with the most supersaturated proteins, we get Parkinson's, Huntington's, Alzheimer's, and then also pathways that are physiological pathways of complex formation. So ribosomes, oxidative phosphorylation, cardiac muscle contraction, the proteasome. The idea being that there's a balancing act in the cell, that you need these conditions. You need to make high concentrations of essentially sticky proteins in order to mediate forming big complexes that are necessary for physiology. But if the balance is thrown off for whatever reason, molecular chaperones aren't there, there are mutations, there are changes in concentration, there's stress, what is physiological can become pathological. That the cell is constrained in the options that it has, and that it can't always manage the difficult trade-offs that are there. And so we come back to this figure that we saw before. Everything in red is actually high on our supersaturation metric. So despite the fact that there are about 100 different proteins here that all seem to be kind of assembled and thrown together for random reasons, actually most of them are here, a disproportionate number of them are here in Alzheimer's disease, possibly because they are supersaturated, or at the very least they share that property. And if we ask, it's not only things, so this is very proximal to the disease cause, things like amyloid precursor protein, which generates the amyloid beta that aggregates, ApoE, which is known to be genetically linked to Alzheimer's, and then things that are more downstream, things that are probably effects rather than causes, like the disruptions in the oxidative phosphorylation pathway and in the endoplasmic reticulum, all of these enriched in supersaturated proteins. And if we ask, is there something in common between Alzheimer's, Parkinson's, Huntington's? In fact, the core overlap between these pathways are particularly supersaturated, so that there may be a certain set of proteins in the cell that regardless of what you hit them with, they're always going to be in trouble. Other things might be a little more specific to the particular diseases, but there's this core metastable proteome that has the chemical principles that put it in trouble. And again, here, proximal to the disease, almost everything is supersaturated. Distal, what we're saying is we can explain some phenomena in terms of supersaturation, like the disruptions in the mitochondrion, which are central to the disease, but other things like inflammatory processes or programmed cell death probably have to do with other phenomena. So supersaturation doesn't explain everything, but it explains a lot of things. And so just to summarize there, the disease-related proteins, they're supersaturated, so are the co-aggregating proteins, so are the ones in aging, so are these physiological complexes and the complexes associated with neurodegenerative disease. And concentration, how much protein you're trying to stuff into that cell, that appears to be the dominant factor that explains this. The cell expresses proteins at exquisite, ridiculously high concentrations, and managing that seems to be a fundamental cellular problem. Now, we we wondered if this is such a big deal for the cell, then there's probably got to be a regulatory phenomenon that helps to manage it. Are there physical principles to how cells turn on and off genes when Alzheimer's sets in? And then, how can we distinguish those transcriptional changes that we see in Alzheimer's from what occurs in natural aging? One of the big questions is, how is Alzheimer's different or similar to natural aging? And can we take, again, a physical principles approach to understanding that? This doesn't look too good, but don't worry about it too much. Basically, we use all of the microarray data. So this is measures of gene expression changes for Alzheimer's patients and control patients that are available from studies in the literature. So about 1,600 microarrays looking at all of the genes in the transcriptome. And we ask, what are the changes between Alzheimer's and natural aging? And can we isolate the aging component from the disease component from the gender component and figure out what are the similarities and differences? And isolating those differences turns out to be really important. This is basically the distribution of ages in the natural aging samples and the disease samples. As you can tell, there is a difference here. The samples that are available from these studies, people who, the brains from Alzheimer's patients tend to just be older. And given that age is the biggest risk factor, you've got to be able to control for that, which is something we've been able to do here by using a linear model for every single gene, where we build a linear model that takes each factor, age, disease, gender, the tissue, so does this come from the hippocampus or the frontal cortex, and isolates it to ask what are the factors that drive each one. And what we see is that there are these distinct but overlapping changes in Alzheimer's and aging. There's a big core of stuff that overlaps in what gets turned down in aging and Alzheimer's and what gets turned up, but most of the changes are different. So these are two very distinct phenomena where lots of different kinds of changes are happening. They do have some stuff in common. And one of the things that they appear to have in common is a response to supersaturation. So if we ask those proteins that co-aggregate in plaques and tangles that we found to be supersaturated before, everything above this line is sort of statistical significance. So the red is what gets turned down in disease, the gray is what gets turned down in aging. And it turns out that a statistically significant, so a disproportionate number of the proteins that clump up with those plaques and tangles, they get turned down in Alzheimer's disease, and at least with the tangles, in aging as well. The idea being that why would that be the case? Well, if concentration is a problem, if you have too much of these proteins around, then one response that might help to ameliorate the extent of toxicity would be to turn down the production of those problematic proteins. And that seems to be what's happening. And we see the same thing if we ask, well, of all of the genes that are turned down, what is the supersaturation level of the proteins that are associated with them? So these are the genes turned down and up in disease and in aging. And as you can see, the things that are turned off have a very high supersaturation level, 10 times the rest of the transcriptome. Again, the idea being that the things that are getting turned down are the ones that have this concentration solubility problem. They're supersaturated. And this may represent a novel strategy for the cell to be able to deal with the misfolding problem that it has just by regulating the levels of the various proteins that exist. And it might be particularly necessary to do this in the disease, because what we see is that in the red again, the disease, the proteostasis components, things that manage the trafficking, folding, signaling that's necessary to keep proteins folded, these are disproportionately suppressed in expression over the course of the disease. And it's been shown, I think there's some sample size issues here, but it's been shown that that's true for aging as well. So the functions that normally keep all of this stuff in balance aren't working as well, especially the folding processes and the trafficking processes, and the membrane is very important in these diseases. And so in absence of a well-functioning proteostasis machinery, the transcriptional regulation that we observe might be necessary in order to keep the cell going for at least a little bit longer. And again, we looked at cellular pathways. Most of them don't change. A few do. And what we find is an exquisite correspondence between the pathways that are supersaturated and the pathways that get downregulated. And the more a pathway is supersaturated, in general, the more it's downregulated. The ribosome, which is a big, highly concentrated, but also very highly regulated complex, is really supersaturated, but doesn't get downregulated. And the ribosome has lots of, because it's so central for the function of the cell, it has lots of special regulation. I think it'd be interesting to explore why this seems not to need the, or maybe it does, and that's a problem, but why does this not get suppressed to the ribosome when everything else seems to sort of fall on this line, where the more at risk it is, the more its expression gets suppressed in the disease? This happens less so in aging. So if we compare basically what gets suppressed in the disease, Alzheimer's, to what gets suppressed in aging, most of the pathways don't correspond. What does correspond, Alzheimer's, which amongst all the neurodegenerative diseases is the one that is most particularly age-associated. So the disease that's most age-associated, the proteins that are associated with it, get suppressed not only just when the disease sets on, but as a person gets older. So there may be an extent to which those phenomena that are particularly disrupted in aging, there's a built-in regulation already. Now, this is actually maybe problematic for the cell, because some of these functions are really, really important. And so you're picking between two bad options. Either you express all of this stuff, and it begins to aggregate, and you're in trouble. Or you don't express it, and maybe doesn't aggregate, but you don't have any of it around. And so then oxidative phosphorylation doesn't work as well. The cell doesn't function as well. One other interesting phenomenon we saw here was that the activation of inflammation, which has always been thought to be associated, a transcriptional activation of inflammation has been thought to be associated with Alzheimer's disease. We don't see that effect. We only see it being associated with aging, which underscores the importance of isolating those components. A lot of the previous studies on that kind of lump the aging in with the disease and don't see the distinction where, as you get older, the coagulation cascade, things that are associated with lupus, so again, inflammation, gets turned on and on and on. There's not necessarily a strong compounding effect where as you get older and you get the disease, it gets even worse. This is mainly just normal aging. So those distinctions and working them out are of incredible value. So again, distinct but overlapping pathways, the co-aggregating proteins are downregulated, so is proteostasis machinery, so are the supersaturated proteins. And the pathways that are downregulated in disease correspond exquisitely well to the pathways that are supersaturated. There seems to be some co-regulation there. And then finally, inflammation, which people thought has been disease-related, really may be about natural aging instead. And so finally, one last story that I'll leave you with very quickly in the last few minutes we have here. We've discussed what happens once proteins misfold and aggregate and how that's associated with a number of different diseases. But right at the beginning of a protein's life cycle, there's an opportunity for it to either fold quickly and maybe avoid some of these phenomena, or to come off of the ribosome unfolded and maybe more at risk for misfolding and aggregation. Now, it's very difficult experimentally to look at how a protein folds on the ribosome as well. It's been done for a couple of proteins. This is an example where essentially as a protein is made, when an enzymatic domain folds up, it enables a cleavage event that you can then measure. So you know that as the protein is coming out, a cleavage has happened, which means that the domain that mediates that cleavage, the protease domain, is already folded. So when biology is helpful, and the particulars of a protein enable it, you can measure this co-translational folding process where the string becomes the globular shape. But when it isn't, you really can't. And so we've taken a computational approach to try to measure or try to estimate when, as a protein is being made, each individual domain that makes it up is likely to have folded. You can think about this as an equilibrium process in which you stop the ribosome at every new amino acid and you give the protein time to fold if it can fold or not. Or you can say, let's take a look at the folding process as it is racing against the translation process. So the protein is being made, eventually it'll be released. At the same time, the domain can fold and unfold. And there's a race going on between them. And this is work that was done with a phenomenal postdoc in our group who's now an assistant professor at Penn State, Ed O'Brien, who came up with this very complicated equation. But the basic principle is that it's a race between the time it takes for a domain to fold, the time it takes for it to unfold. We can estimate this using the size of the domain and the structural classes, an alpha domain, a beta domain, or an alpha-beta mixed domain, and the time that it takes for the protein to be made. So if the protein takes a long time to be made, there's more opportunity for the domain to fold. It's very quick, then unless that domain folds really fast, the protein will be released in an extended conformation. And we can estimate this time using some models that exist in the literature as well. And basically what we say is that we call a domain autonomous folding units that are larger than 50 residues. They've been defined in various databases. The folding, we only say that a domain can fold after it's fully synthesized. This is probably true, but there may be exceptions. We consider it a two-state process. So basically, you're either unfolded or you're folded, and there's some probability of each state. The translation rates we're estimating based on the individual DNA and RNA sequences. And we say that codons of a given type have a fixed rate, although there's some evidence from our group now that that might not necessarily be true in all cases. And we say that the rates are governed essentially by how well the machinery of the cell can kind of match on to onto the right. So whatever the tRNA, which is carrying the amino acid, which needs to get synthesized, how well that can match to the mRNA molecule that carries the information about the instructions for what needs to be made. And then finally, we focus on things that are cytoplasmic or nuclear localized because our model for the ribosome and translation and folding off of it only really applies to free ribosomes and not ones that are stuck on the endoplasmic reticulum. And then finally, co-translational folding. We say that a domain is co-translationally folded if we predict that its folding probability is greater than 50%, and that at least one, and we say a protein is co-translationally folded if at least one of its domains gets folded in that way. So what do we see? Well, when we put all of this information together into that chemical kinetic equation and generate one of these co-translational folding curves, there are some domains that are fast folders. And regardless of whether you stop the ribosome or you let it go straight through in translation, you get lots of folding. In other cases, the speed of translation is really limiting. And so something that could fold if you gave it enough time just will not fold in the time that it takes for the protein to be fully synthesized, which is not a long time in some cases. So these kinetic effects are really profound. And in some cases, you're talking 40 to 60 amino acid delays in where you would expect the domain to be able to fold if you gave it all the time in the world versus where it folds if things go on at the natural rates in vivo. So there's a certain metastability there in the sense that these domains cannot always achieve their thermodynamic equilibria given the constraints of the kinetics. And the domains that are most at risk are large beta-rich domains where we can see that the post-translational folders, so the things that don't fold on the ribosome itself, tend to be larger than the co-translational folders. The post in green, the black is co. And then, as well, the domain. If we look at the domain classification, the co-translational folders tend to be the post-translational folders tend to be disproportionately in beta domains. And similarly, if we ask, what's the delay like? So this delta LM value again, how long? What's the difference between what you would expect if you gave it all the time in the world versus only as much time as it gets in the cell itself? We see that larger domains tend to have bigger delays, and the beta domains tend to be a little bit more delayed than the other domains proportionally. The alpha beta ones are also a little bit problematic. So there's widespread co-translational folding. This is in E. coli. The majority of E. coli co-translationally folded domains have these delays, which introduce a potential metastability. And for 20% of all of the cytosolic proteins in E. coli, this can actually be so profound that it switches what you would think is a co-translational folder to a post-translational one. And you can imagine that the various changes and conditions in the cell tweak and push this further so that there can be more disruptions in stress and disease. And again, the large beta strand rich domains are particularly problematic. And I will zoom through this in just a couple minutes here. But basically what we wanted to do was to work out which physical determinants mattered most for co-translational folding, especially as you move from the single cellular bacterium E. coli that we looked at to eukaryotes like us. But not quite like us, we looked at yeast, which is also one cell. But it's a nice comparison because as you shift from a bacterium to a single-cell eukaryote like yeast, there's an incredible increase in the complexity of the proteome. The proteins are more complicated, they're larger, and the folding problem is harder. So what we wanted to know was what underlies that problem. So we compared a bunch of proteins and domains in E. coli to as many as we could in yeast, given that we had the structural data necessary. And what we find is that there's more co-translational folding predicted in blue in yeast than in orange in E. coli This is both, so here we're comparing 37 degrees in E. coli, which is the temperature it likes to grow, versus 30 degrees in yeast, that's the temperature yeast likes to grow. And what we find is, yes, this may be a way of essentially managing the increased complexity of the yeast proteome, because you have a significantly, a small but significant increase in the amount of co-translational folding that occurs. So you have a more complicated protein, you try to fold it more. But the very features that make the yeast proteome more complicated may also be contributing to that co-translational folding. So first, let's look at temperature. At every given temperature, if we control for that, right, that could be a big phenomenon in the kinetics here, yeast is still more prone to co-translational folding than E. coli is. So it's not just that the temperatures are lower, things are slowed down, everything can kind of get, the folding can kind of get sorted out. In fact, even at the same temperatures, yeast folds more co-translationally than E. coli. And what about these timescales? So this is translation, this is folding, this is unfolding. How do these fundamental timescales, which ones contribute, which ones distinguish E. coli from yeast? It could be that we see more co-translational folding in yeast because folding is just faster in that organism. It could be because unfolding is slower. It could be because the translation rates are so slower in yeast than E. coli. Which ones of these matter? We looked at this by essentially comparing what are the differences at the optimal growth temperatures in the timescales between unfolding, folding, and translation between E. coli in orange and yeast in blue. It's hard to tell here, but this is actually a significant difference. Probably a better way to plot it. But basically, unfolding time doesn't matter. It's the same between the two organisms. Folding time is 3 times faster in yeast than it is in E. coli. So that seems to be in play. And then the translation is 7 times slower in yeast than it is in E. coli. So that also seems to be in play. Those two things seem to matter. If we now control the temperature, this faster folding time in yeast really seems only to have to do with temperature. So the lower temperature makes, it decreases the tau F value for yeast, but at the same temperature it's not an issue. Whereas the translation rates, even controlling for temperature, are different in yeast than in E. coli. So it's not just an overall property, but the individual properties of the codons. And so we wanted to look into that a little bit more. We figured the translation times depended on 2 things. How long do you have to translate in terms of what's the length, so how many amino acids still need to be translated, and then how much time does each amino acid take to be translated? Those are really the 2 underlying phenomena. And what we find is that post-domain length, so the length of protein that's left to be translated after you make a domain, is almost perfectly correlated in both E. coli and yeast with this translation time value. So basically what we can say is yeast proteins are big, and that makes them complicated. But that same complexity facilitates more folding because it essentially gives you more time on the ribosome to co-translationally fold. There's much less of a correlation with this post-domain tau A value, mainly because the post-domain lengths are so large that this is a weak correlation with the average time that it takes each amino acid to be made. And the separation really in post-domain lengths is really significant between the 2 organisms. And so is the post-domain tau A. Basically, yeast uses both options. It both slows down each amino acid, and it has a much longer length. And together, those 2 give that big 7-fold difference in translation times. But we did see something weird right here, which was that there's a set of yeast domains that operate, looks like on a different principle. They're sitting on a different line. And we wanted to know what was going on with those domains that have a really fast translation time. Those domains sit in proteins that are really important for translation and for enzymatic reactions. And they're extremely highly abundant. So the idea, and this is consistent with things that have been seen in the literature, is that the really fast domains, the really fast proteins that are translated in yeast, tend essentially to come from highly abundant proteins that need to be manufactured quickly. But I'm going to just focus in on this. If we look at the entire proteome, so not just the stuff that's in our study, but all of the proteins, what we see is that at the optimal translation rate, most yeast proteins are sitting around here. They're pretty slow compared to E. coli proteins. But there is that group, this long tail, shapes are different between these 2, that goes really fast. Those really fast proteins seem to sit at a limit, a speed limit for translation, where you just can't really make things a lot faster than this level. Otherwise, you'd expect there to be another tail for E. coli that goes almost to 0. But the eukaryote, while it needs to slow down things in general, has some proteins that need to operate the way prokaryotes do, need to operate at high speeds. Those tend to be highly conserved, meaning their sequences are very similar between E. coli and yeast, more than for the rest of the proteome. And in fact, the co-translational folding properties in general seem to be highly conserved. So things that have either, that are both co-translational folders in yeast and E. coli or not, the extent to which that's the case seems to be related to how much the sequence is conserved. So despite the fact that you change everything else about the organism, the translation rates, the size of the proteins, the folding rates, the unfolding rates, if the sequences are similar between E. coli and yeast in a given domain, then the chance that they behave similarly and whether they fold properly also seem to be similar. The idea there being that there's been a lot of other evolution to accommodate the same outcome of either folding on the ribosome or not, of either being metastable in that way or not. And then the final thing we looked at, domain size and class. So we had this puzzle, which is that beta domains are tough to fold. Everyone knows that, especially big ones. But in fact, if we look at the amount of co-translational folding that we see, the alpha-beta domains, which ought to be intermediate between alpha and beta, actually are worse off than everything else. And that's true in E. coli and yeast. But what we wondered is whether this had something to do actually with size. So is it the case, perhaps, that the alpha-beta domains don't fold so well because they tend to be bigger? And so we defined this, this metric, which we call the capacity for co-translational folding. And it's pretty simple. All we're saying is, if domains in general, the median size of domains, if the domains in general tend to be big for a given class, then the capacity for co-translational folding really needs to be balanced against how large a domain can fold well in that organism. So this is the midpoint folding probability for all of the beta domains in E. coli. You can say, well, at around 225, about 50% of the domains co-translationally, or there's about a 50% chance that a beta domain in E. coli co-translationally folds. But the median size of a beta domain in E. coli may be less or greater. So how do those two values compare? And what this lets us do is to come up with a nice metric that correlates well with these fractions of co-translational folding. So the larger this mid-F value is compared to the median value, the more likely we are to see co-translational folding. And this is shown as one of the reasons why, one, the alpha-beta domains are problematic, and two, why the beta domains maybe drive the evolution of co-translational folding capacity in yeast. So to look at the, we'll look at the alpha-beta domains first. Basically, even though the alpha-beta domains have a higher size mid-F than the beta domains do, so they should be better at co-translational folding overall, their domains tend to be much larger. So the ratio here is worse for alpha-beta than for beta. Basically, there isn't enough excess slack in the alpha-beta domains to make up for this much larger domain size. And so the alpha-beta domains tend to be more problematic to fold. However, when we compare yeast to E. coli, we see that while in most cases, at the normal growth temperatures, the same sort of capacity for co-translational folding exists in yeast, say around 1.95 in fact, it's even better in yeast, so about 1.95 at 30 degrees, in E. coli about 1.7, and if we look at the same temperature, about 1.7 for the alpha domains at 37 degrees in yeast, and about the same thing in E. coli. The beta domains don't operate that way. At the same temperature, the beta domains in yeast do much worse, or significantly worse, than do the alpha domain, or than the beta domains in E. coli. So there, what we're saying is that in yeast, beta domains, if you grow the organism at the same temperature, they're not going to have as good a capacity to co-translationally fold, even though overall the organism can do co-translational folding better. It's only when the temperature is dropped from 37 degrees to 30 degrees that that co-translational folding probability comes in line. So essentially, some of the strategies that the organism takes, for example, having a lower temperature, doesn't really make a difference to improve the quality of co-translational folding for alpha and alpha-beta domains. They're already at the same level, even at the same temperature in yeast, despite the more complicated proteome. But the beta domains
really can only fold at the same level, can really only reach the same level that the more primitive organism gets when you drop the temperature. And what that suggests is that there's essentially been an expansion in the size of beta domains in yeast. That expansion has pushed the co-translational folding machinery in order to be able to accommodate these very large beta domains. So it may not matter what changes have happened to the alpha or the alpha beta domains or the smaller beta domains. The limiting factor that has driven the evolution of co-translational folding in the eukaryote versus the prokaryote is accommodating having these big, tough-to-fold beta domains that have a low co-translational folding capacity, except at the optimal growth temperature. And so to just summarize there, the timescales for folding seem to be important because of temperature. That helps with the co-translational folding. The timescales for translation also important, and in fact, the complexity of the organism contributes to it. But there's special regulation for yeast in a certain set of proteins that are highly conserved. And finally, by looking at domain size and class, we're able to identify what the real constraints are on co-translational folding, so that even though overall the proteins and the proteome seem similar, there's a small subset of large beta domains in yeast that may really contribute to the kind of metastability that you could see in the organism. And it's not surprising then that large beta domains tend to be the ones that aggregate and misfold, not only in disease, but in widespread aggregation. And so we see that there's this greater co-translational folding. It's driven by temperature and by the structure of the proteins themselves. The eukaryotic translation machinery is slow, and that helps. But there is differential regulation there. Co-translational folding is a conserved property, and large beta domains seem to be a big constraint. And with that's basically what we've been able to achieve is I don't know what this is, but let's go back. What we've been able to achieve is to look at these individual time points where there's a risk for misfolding and aggregation, and to work out the kinetics and the thermodynamics that underlie the basic principles for why hundreds, if not thousands, of proteins can be disrupted in stress, in aging, and disease. And what I hope that we're able to start to get at is an answer to this question of how the complexity of human disease links up with the complexity of cellular biology. And that maybe it's not a mystery and a black box, maybe it's not insoluble, but that in fact, simple chemical rules help to explain how you go from translation rates and aggregation and co-translational folding to the real devastating, tragic consequences that patients face. And maybe, just maybe, we can use that information to improve diagnostics and treatment based on the actual chemistry and pathology of the diseases we're studying. And with that, there are so many people to thank. So many people from the lab who are here today, from Chris's lab, from Michele's lab, collaborators here and around the world. Of course, I've got to thank Rick. This is a somewhat less silly picture we had together. Chris and Michele, who have been phenomenal advisors, who taught me so much about science, so much about thinking about complicated problems. It's been tremendous fun, and I hope that our collaborations will continue with everybody in the lab and with them. I'm, of course, very thankful to lots of people who are here, who helped me to get my fellowship to go to Cambridge, who advised me from undergrad onwards. Of course, I'm incredibly grateful to my parents and my brother who are here right now, to my second mom in Chicago, Carol, and Ron, who's her late husband and my second father in Chicago, really, to friends, some of whom are not here, and family, some more friends who aren't here, and of course, we dress like garbage bags sometimes. A few friends who are here. So John, thanks for making the trek from the University of Chicago. I think, Taylor, do you remember this? This is a good time. We were right at the beginning of the MD-PhD program, so we were a bit younger and maybe less foolish, actually, not more. And, of course, very old friends from undergrad. Alex, thanks for making it out. April, thanks for being here. Of course, I'm also grateful for the MSTP, so I'm glad James was able to make the trek down. I didn't have a picture with you, Chandler. Otherwise, you got to send me the ones we took recently, but it's great to see you here as well. And thanks to everybody for being such a big part of my log and tortuous journey, which continues on Monday.
Yeah.
Thank you.
Before questions, we will have reception, food, party shortly after. Probably we'll continue talking, and we'll set up at that point. Some questions?
Well, is there a correlation between those proteins predicted as saturated or co-translational?
So we don't have good information on that yet because our estimates for co-translational folding are coming from lower or are for lower organisms. What we'd like to do is to be able to take structural information for C. elegans and humans and look at that. One thing that we do know is that the supersaturation phenomenon seems to have different rules in rapidly dividing single-cell organisms from what we see in multicellular organisms. So we really need to make a better comparison of that. So we're not sure yet. We're working on it.
Do you think then, I mean, if all of these diseases are tied together, and towards the end of your talk, you talk about how slowing down the rate of translation gives them more time to fold and avoid some of these aggregation problems. Do you think that could be a common therapeutic approach to just slow down the ribosome just a little bit?
If you could find a good way of titrating that, then yeah. So that's the challenge, right? So there's been some evidence that if you shut down translation, which you can do easily, you get a short-term resolution of some of these problems. But over the course of a few years, I think shutting down translation may cause some other problems for the cell, some minor ones. But maybe if you could slow it down, that would be great. I just don't know if that's an easily druggable thing. Who knows?
But just to study the basic biology, do you think adding in an egg of codons that they move a long time to translate?
There's some of that work has been done, and so if you so some of this has been done on individual sort of model proteins to see whether you can increase or decrease co-translational folding using the tag. Now, what I don't know if that's ever been done with a disease-related amyloid protein. So that would be a really interesting experiment.
Where does all the data come from that decides, for instance, just an example, the difference between the alpha helices and the beta sheets and the alpha beta proteins?
Yeah, so
is this all annotated by
yeah, so most of the data comes from either with the alpha and beta domains, we define what the domains are using the stride algorithm. So we take we take high quality crystal structures or NMR structures, and then we look at the coordinates, and that allows us to sort of determine whether it's likely to be an alpha or a beta domain. But then the sizes of so then we can get the sizes of the domains as well, because the domains themselves are defined in scope, CAF, domain parsers, so essentially these databases that use structural information in addition to homology information in order to say, well, this is a domain in this protein and this isn't. So it's a combination of algorithmic approaches and database approaches.
So this wasn't done by hand, some of it automated.
So most of everything was automated. Now, scope and CAF, a lot of that is done by at least some chunk of that is done by hand. So there's, of course, a question as to what the fidelity will be of any algorithm, right? There's going to be some limitation on that. But when we're talking about looking at thousands or tens of thousands of PDB structures, I like the automation.
Prajwal, when we first met a while ago, we had considered that given genomic information, moving towards precision medicine or personalized medicine, could one look at each protein encoded from genomic sequence and predict proteostasis index? Likelihood that you encoded a healthy proteome versus possibility of risk factor.
So
how much progress have you made towards that?
A little. Earlier this week, I was having a chat with someone. Apparently, you can do a pretty minimally or non-invasive nasal lavage that allows you to extract neurons. and there's no real pain to the patient. You get a few neurons. Of course, olfactory disruption is probably the first thing that happens in, say, Alzheimer's disease. With single-cell transcriptomics, you could then look at the expression levels and the sequences from those neurons in those individuals and ask, is this person's transcriptome more at risk than someone else's? And potentially, at some point, you might even be able to look at particular markers. But at the very least, you could do the transcriptomics on it and then say, you've got this set of mutations, and you also express things at too high a concentration. I'm a little concerned about you. Versus, oh, you're great. Everything is sort of low expressed and well, very soluble. So I think that that's something where we've got sort of the technical things in place.
So
We
this
need
is the
samples.
reason why you're going back in the clinic, so you can finish this part.
So I can go around to people with a little bit longer and get their cells, yeah.
Okay. Let's all thank Prajwal for a wonderful lecture.
Undergrads here keep going.
If anyone is still there, goodbye. Thank you.